Good morning, and thank you to be here to the Women Talent Program final event. So tomorrow is 8th of March, which is the International Women's Day, and from ICN2 we want to join and to raise awareness of the persistent gap which is existent in different contexts and in different aspects in our life. Indeed, science is not an exception. In almost every research institution, female representation goes down as the scientific career advances. And the barriers are diverse. So there are cultural barriers related to personal attitudes. And indeed, the point is not only the cultural barriers, but also the political aspects. There are lack of policies that allows a better work-life balance. There are lack of regulations that guarantees gender parity in selection process and decision-making bodies, but there is also women's self-empowerment. We tend to think that we cannot do things as men do. We are often very self-demanding and often making the mistake to take control over all personal and professional aspects in our life. And this is an error, or this is something that it has to be changed. And it has to be changed by all the community, men and female women that we are here. From ICN2, we are committed in establishing policies and action and corrective actions to try to amend this gender gap. One of the most relevant corrections has been during the last year the Women Talent Programme, which is an initiative which is included in the framework of the Severo Ochoa Center of Excellence 2018-2022. The Women Talent Programme has been aimed at supporting women scientists, basically from the early research and intermediate research career. Indeed, what we are doing, or what we did, is to promote the best price of the thesis, the best price of the paper, and the best on some projects for the postdoc grant and the senior grant. Let me put some numbers in this program. So in total, we have 3 calls of thesis, and we evaluate 57 theses, 2 granted and 1 to be granted during 2022, which will be at the end of this event. For the papers, we had 3 calls, and we evaluate 76 papers, 5 granted and 2 more to be granted during this 2022. And 2 calls of projects, 24 proposals, and 2 projects granted for a total of 30,000 euros. Indeed, what I am doing now here is to take over the work done by this commission for a long time. I want to thank all the members of the commission, Nuria, Belén, Julio, Alex, Marius, and many others. But among all, I want to thank the work done by Neus Domingo, who is now in the States working, and I think that she's connected now, who did a lot of work to make possible this Women Talent Project final event. With this, I just want to emphasize that the gender balance is not an option, it's a must, and we are all participants to make it possible. With this, I just want to welcome again to the Women Talent Programme final event, and I wish you all the best for tomorrow, which is the International Women's Day. Thank you very much for being here. Good morning, Sonia.
Yeah, good morning.
We will welcome our first awarded researcher, who is Adriana Figueroa, for that she will introduce her project D-Spin from 2019, that's terahertz emitters with spintronic devices. So Adriana, the floor is yours.
Okay, now, all right, thank you. Thank you very much for the invitation. My name is Adriana Figueroa. I used to work here at the Physics and Engineering of Nanodevices group, led by Professor Sergio Valenzuela. And in 2019, I applied to the Women Talent Program and I was successfully funded this project, which is called the Spin, terahertz emitters with spintronic devices. which is a very nice project that we started then. And today I'm just gonna do a briefing of the project that we started then, and I hope you enjoyed it. So it will be a short introduction about it. So I just wanted to start by introducing why terahertz. So in terms of why are we interested in terahertz emitters or in terahertz technologies, this is a technology that is being used nowadays in several areas, in several sectors. So they go from medicine, imaging, fundamental research, or ICT, information and communication technologies. And the idea here is to develop not only emitters, but also detectors that are compact, that are reliable, that are fast, and they are low cost, right? Nowadays, the emitters that are being used are rather bulky and expensive. So it is important to work on strategies to develop novel technologies that could replace these terahertz emitters. And in terms of emitters that make use of solid state devices, nowadays the technologies are based on insulators and semiconductors. And in the recent years, there has been efforts put into developing technologies that are new or novel, that make use of novel effects, novel phenomenology, that, for example, it has been demonstrated that the use of spintronic devices that incorporate magnetic metallic thin films with ferromagnetic materials, at the interfaces there are phenomena that could be used to develop devices, terahertz devices that are very efficient and very powerful in terms of broadband. They have very nice capabilities that we can use to replace the novel technologies in terahertz. So this is why we were interested in working in this area, in this sector, because we our group is an expert in spintronics, and then we wanted to make use of the knowledge that we already have and the devices that we could develop to explore this area of terahertz emitters. So in particular, our group work with novel materials like topological insulators or 2D materials or quantum materials. And in the last years, it has been demonstrated that thin films of the incorporating, or like heterostructures incorporating thin films of topological insulators, indeed, in fact, can emit in the terahertz range. So, and they are very efficient as compared to single metallic and ferromagnetic interfaces. For example, here there is a work performed in 2018 that show a very good an enhancement of these signals that we can obtain with such materials, with topological insulators. So this is why we decided to investigate in this area, and then to make use of films or devices that we already have, or like heterostructures that we have already characterized for spintronic applications, but now to explore their performance as terahertz emitters. So in this sense, we use samples that we grew by molecular beam epitaxy in our group, in the physics and physics and engineering of nanodevices group. In order to explore the terahertz emission, we use setups and instrumentation that was already available at the photonics and phononics lab led by Professor Clivia Sotomayor. In particular, we use this setup, which is called the ASOPs, the asynchronous optical sampling equipment. And for the measurements and for the analysis of the performance of these materials, we made use, took advantage of the expertise and experience in terahertz spectroscopy provided by the group of class Gianterrogi. So our project was designed in a way that it was synergetic, so that we could combine the expertise of these three groups in ICN2, the physics and engineers of nanodevices, the ultra-fast dynamics in nanoscale systems, and the phononic and photonic nanostructures. So it was a very nicely designed project that could integrate and collaborate between these three groups at ICN2. So this is the introduction of the project. I mean, what we were aiming at was to develop, or like to investigate and develop to investigate the performance of these heat-rate structures integrating topological insulators and magnetic materials, and to evaluate the response as terahertz emitters in the, to investigate how they emit in the terahertz range. So in order to do so, the first thing that we did was to grow samples and to make sure that these samples were, have the same behavior as we have already demonstrated in the past for not only for the spintronic, for the spintronic properties, but also like the structural and electronic properties. So we grew them in the molecular beam epitaxis system at Penn. Then once we had the samples, we also, in addition, we also needed to make sure that the setup available at P2N was working properly in the terahertz range. So the thing is that this setup has the capabilities to work not only on the terahertz range, but also on the gigahertz range. And this system was only used in the gigahertz range so far. So what we needed to do was to do the commissioning for this A subsystem to work, to perform measurements in the terahertz range. And that was successfully done. So it was set up for these measurements. So now that we have the two components, we put them together and we tested our samples, right? Not only the samples with TIs, with topological insulators, but also some reference samples to make sure that the setup was working for such, to allow for these terahertz emission measurements. However, unfortunately, what we, despite the efforts that we put and despite the tests that we performed, unfortunately, we didn't get any terahertz signal, neither on the reference samples nor on our samples with topological insulators. But we think, or like we found some results for, some ideas why this could be. We think that the gigahertz repetition rate was too fast to deliver enough power for our samples to emit on this range. So we went to the contingency plan. So we had a contingency plan to test the response of our samples in a different setup. So in order to do that, we explored the possibility to collaborate with the group of Professor Mezziani in the University of Salamanca. And so what we did was to get in contact with them because they are experts in terahertz spectroscopy as well. And then what we did was to test our samples there. So far they have our samples. Unfortunately, they have not seen terahertz signal yet. But what we are doing now is to explore, to try to test their setup with a reference sample that we know that emit in the terahertz range, which is provided by Professor Kampfram from Berlin, who has already demonstrated that this sample they emit in the terahertz. And just to summarize, what are the outputs of the project so far? So we have launched terahertz measurements at ICN2. We have demonstrated a nice synergetic and collaborative interaction between the groups between these three groups at ICN2. We have applied to further funding, but we have not been successful yet. And this project has helped me a lot as a PI. I can talk about it later if you're interested. And we have established, this project has allowed for collaborations with the University of Salamanca. And with this, I just finished the briefing of my project, and I want to thank everyone for your attention, and also to the Severo Ochoa Program and the Equal Opportunity Committee for for funding this nice project. And this is work in progress. So I just want to say that stay tuned, because we can tell you more about our results later. Thank you.
So Sonia, the floor is yours.
Okay. Thank you, Adriana, for the nice presentation of the project. Yeah, I see there are no open questions. So, well, I guess we will introduce our next awarded, who is Maria de la Cruz Cardenosa. And she was awarded a project called ANELOS, which is Alzheimer diagnosis by neurofilament light detection employing optical silicon sensors. So, Maria, the floor is yours.
I've been working in the Nano B2A group led by Laura Lechuga, and my project basically and our group is dedicated to the development of these kind of sensors. So why this project? And why Alzheimer? As you know, Alzheimer disease is a progressive, like neurodegenerative disease. It's caused by these plaques that are deposited in the brain, and so the neurons start to die. And it's a progressive brain degenerative disease. And why to study it? Because it's the main cause of dementia in older adults, and also it's the top 5 cause of death in adults. So currently the diagnosis of this disease is based on the sign and symptom evaluation by doctors, as well as other imaging tests like MRI, CT scans, and PET. But there are some drawbacks with this, principally the infrastructure and equipment cost of doing this kind of tests. Then they require for the patient to be hospitalized during long times, and also like the mild brain damages cannot be detected, so sometimes it's very hard to evaluate the condition of the patient. So now people are trying to change from this imaging test to biomarker associated test to diagnose AD. So when in this disease, there are a lot of biomarkers that can be detected in blood or in the CSF, the cerebrospinal fluid. Most of them, they can be related to these plaques of amyloids that are formed in the neuron, but as well, there are other markers related with how they, when the neurons break, they release these filaments, neurofilaments, and also there are other proteins related with the inflammation of all the glia that is in the brain. So why rely in this kind of biomarkers? Basically because there are some kind of advantages. So they don't require so much cost in all the instrumentation. Also the patient, it doesn't need to be hospitalized. So you just can withdraw a sample of blood or serum and detect, cerebrospinal fluid and detect the biomarkers in them. Like this kind of test could be more sensitive. By detecting these biomarkers, we could detect mild brain damages. As well, they can give us other kind of information related with the disease. Indeed, my project is focused to detect one of these biomarkers, the neurofilaments. So it's a very these biomarkers has been discovered very recently, and has been a lot of attraction to it because it's related to any kind of brain damage, and it's very selective of the brain the damage that is happening only in the brain. So these filaments are found in the axon of the neuron. So when they break due to these plaques that are causing, these neurofilaments are released into the CSF, the cerebrospinal fluid, and then they are released, they pass the brain and they go to the circulation, the blood, and they can be detected in the blood. So actually has been a lot of correlation between Alzheimer's disease progression and an increase of this biomarker in blood. And not only is related to Alzheimer, but it could also serve as a biomarkers of other neurodegenerative disease like Parkinson, as well as other as brain trauma, ischemic strokes, etc. So that's the reason we try to focus to detect this biomarker by using optical sensors, the sensors that we develop in our lab, the bimodal waveguide sensor. So basically, we will put some antibodies in the sensor to try to detect these neurofilaments in CSF as well as serum. So I'm sure many of you have heard the main sensor, that silicon sensor that we developed in Laura's group. So they are called the bimodal waveguide interferometers. They are made of silicon, and they have these structures that they are called waveguides. So we can couple the light on the waveguides, and then there is, well, a step junction. We have these two modes of light that generate interference pattern. By placing like antibodies here on the surface, we can sense everything that binds or happens in the sensors, because they are very sensitive to any kind of refractive index changes. So by monitoring whatever happens in the surface, let's say like the binding of the target we want to detect to the antibodies like very selectively, we can detect any kind of molecules that bind to the surface. So they are very sensitive. They are label free because they measure the refractive index changes, and also they are like low cost because they can be made by semiconductor fabrication techniques. So with all that, my project basically consisted in the evaluate like different antibodies. So I have to test like from different companies antibodies to test which one offer me the best affinity. Then optimize all the sensor surface functionalization as well to the detection of the neurofilament in buffer. Then I went on and tried to detect the NFL in complex matrices like serum and also cerebrospinal fluid. And finally, something that we still need to do is the detection of the NFL in patient samples. So some like CSF sample can be acquired also by companies. So for the NFL detection in buffer, we had to optimize basically the optimization we have to do for any kind of biosensor. First, we need to improve the chemistry of the surface, like what kind of reaction allow us to couple our bioreceptor, the antibodies to the surface. Then check how much antibody we need to put so that we can detect like very sensitive. Then we need to block the surface to avoid other non-specific interaction, and then we need to check if the buffer is okay for the target to bind to the antibody. So after carrying all that optimization, I was able to perform some calibration curves. As you can see here, this is the signal of our sensor after like adding different concentration of NFL, and also adding like, you cannot see here, but this is like a non-target protein, so there is not, I mean, the antibodies were very selective for the NFL. Then we performed the calibration curve to detect our limit of detection of our device as well as our limit of quantification. So with the assay, we saw like good reproducibility and specificity for the NFL protein. We were also able to reuse the same chip by flowing like a basic solution. So the filaments, they were like, they have like flowing away, and so we could reuse the surface again. And also we checked that the sensitivity was in the low nanogram per milliliter. With all that, we decided to move to serum because the result seemed okay. But when you put the serum on the device, I mean, serum has a lot of protein, a lot of lipids, like a lot of molecules inside. And beside the blocking we put on the surface, the serum was also binding a lot. So I decided to employ another like a sandwich assay, like using secondary antibodies and nanoparticles to see my proteins beside all the other proteins that they were on the surface. Like here, like I have the first assay with the neurofilaments binding to my first antibody. Then to recognize if the neurofilaments were there, I used a secondary antibody that was biotinylated and streptavidin nanoparticles to see if the neurofilaments were binding. So with that, very recently, I was able to see some changes. So here you can see the blank when I don't have like a NFL in the surface, but when I have NFL in the surface, I was able to obtain some signals. I'm still doing the optimization and carrying on some calibration curves to see if the results are reproducible. So later on, the idea will be to make these calibration curves and try to measure the NFL also in the serum from the patient with Alzheimer's. So to conclude, as I told you before, so the ANELOS project has aimed to detect this neurofilament light in patients with Alzheimer's disease by using the bimodal waveguide sensors. I have tested different antibodies, and then doing the optimization with the ones that work best. And now I'm working in the NFL detection in the serum. And with that, the future work will be like optimized detection conditions in serum and also the validation of the sensor with patient samples. So of course, that would be. I really want to thank the Women Talent Project, the Commission, for giving me the award and letting me carry out this project. As well, I want to thank my group led by Professor Laura Lechuga and all my colleagues from my lab, as well as the ICN2 and the BIS, because I'm under a Provis fellowship, so as well I need to thank them. So thanks so much for your attention. If you have any question, I will be happy to answer. Thank you.
It's more on curiosity. So I've been working for a while with Alzheimer, and I have what I know is that the plaques and the formation of the plaques and the filament, it's more not a cause, it's a consequence of Alzheimer. Could you please comment on it and how your work it can be adapted to? I mean, it's not that Alzheimer is caused by the plaques.
Yeah, it's not. It's a way I need to kind of diagnose Alzheimer, maybe at earlier stages. When maybe you can do something, it's not a cause, but it's more a consequence, a way to diagnose the way the patient is. And if the patient needs rehabilitation or something, to kind of stop the progression of the disease.
Okay. Thank you very much. It's very, very interesting.
Okay, I think Sonia can help us if we have any question from the chat.
Yeah. Hi, can you hear me? Okay, yeah. So there's no questions in the chat now, but I have a curiosity, the working principle is very interesting and you made very impressive advances, but what does these sensors need to be applied in the clinical tests in real life?
Once it's developed, it's very straightforward to use. You just need to inject your sample. But right now, we're trying to integrate it. Does it answer your question?
Yeah, so I don't know if I heard everything. So in terms of the selectivity, and it's already advanced enough to be applied in the clinical test?
In terms of the sensitivity?
Yeah, and selectivity.
In the selectivity, yes. The sensitivity, we might need to improve a little bit because this protein is in a very low range. But yeah, we are on the range more or less.
Okay,
and the
that's very
selectivity is
promising. Good.
Yeah.
Thank you. Okay, so yeah, there's no more questions coming in the chat. So
thank you.
Thanks for your presentation, and we will go ahead and introduce our last project awarded researcher, who is Mariana Sledzinska. And she will present her project Pencil, that is phonon-electron coupling in twisted 2D layers under mechanical strain. So,
Okay.
Mariana?
Yeah, thank you very much, Sonia, for the introduction. Let me share my screen. So the phonon-electron coupling in twisted 2D materials under mechanical strain is the project that I was awarded last year. So the results that I'm going to present to you are all rather new. And I will start with a brief introduction. So in the field of 2D materials, they possess not only extraordinary electrical, optoelectrical, or thermal properties, but they also possess unique elastic properties. So for instance, graphene is the strongest material in the world, with the Young modulus of about 1 terapascal and the intrinsic strength of about 130 gigapascals. Then another, the second mostly studied material, MOS2, has the Young modulus of about 270 gigapascals. So that's comparable to steel, and the critical strain somewhere between 6 and 11% for single and B layers. That makes these 2D materials perfect candidates for flexible and stretchable devices. But not only that, we can also see that strain in the 2D materials can also lead to generation of some unique properties. For instance, in the monolayer WSE2, which is a transition metal dichalcogenide, we can observe single quantum emitters in the strained regions. So the whole new field that has emerged recently called straintronics is related to strain engineering to tune the electronic properties of these materials and explore new quantum states in condensed matter physics. In line with this introduction, with my project, I would try to answer some really fundamental questions. So first of all is how much strain is actually transferred from the flexible substrate to the 2D material or to the 2D layer. So this is actually crucial for creating 2D flexible devices. And then how much strain is transferred from one layer to another, depending on their relative orientation. And this question stems from the fact that all these 2D materials, especially the TMD, such as MOS2, they have been used for many, many years as solid lubricants. And they have this application due to the fact that the layers can slide relatively between each other. So then finally, I'm going to see how strain affects electronic and phononic properties of the 2D materials. And this project aims at answering this question by studying phonon and excitons and their dynamics in the CVD grown semiconducting single layer MOS2 and its bilayers with twisted angles on flexible and stretchable substrates. We can briefly see the schematic of the project in the image. So first of all, we are aiming at growing high quality MOS2 monolayers and twisted bilayers by means of CVD. Then we would transfer them on the flexible or stretchable substrates. And then we would measure the optical phonon and exciton frequencies and lifetimes in strained 2D materials, and also the effects of interfaces and stacking. In this project, we will use mainly the optical techniques, which are non-destructive, such as Raman spectroscopy, photoluminescence, and ultra-fast pump probe measurements. The project can be briefly divided in three, let's call them work packages. So in the work package 1, in which I'm collaborating with Maria Jose's plan view, it's on the TMD's growth and transfer using CVD method. In the work package 2, I measured the exciton phonon interactions in strained TMDs and heterostructures. And then finally, in the package, in collaboration with the group of class, we will measure the exciton dynamics in the strained 2D materials. To start with, together with Maria Jose and two of the students that work with me, Peng and Eulalia, we have worked on development of the CVD growth for sulfur-based TMDs. In the image, you can see the schematics of the CVD growth that we use based on the solid precursors. And first of all, we have successfully managed to grow MOS2 monolayers on silicon oxide. But very recently, for some reason, we were having a lot of problems with MOS2 synthesis. So in the recent month, we have moved to WS2 synthesis, which apparently turned out to be more successful. So we can see either the single layer triangles, or we can also see that we have bilayer and tri-layers with different orientations, okay? So in the rest of the talk, I will just concentrate on the WS2 monolayer optical properties. First of all, in the photoluminescence spectrum, we can see the very pronounced A peak, which corresponds to the neutral exciton related to the direct band gap of the monolayer. WS2, in the same way as other TMDs, MOS2, and so on, shows the transition between the indirect to direct band when we reduce the number of layers. Similarly, for the Raman spectra, we can see the two characteristic peaks, the A mode, which corresponds to the out-of-plane vibration, and the E mode, which corresponds to the in-plane vibrations. And in order to be able to apply control strain in 2D materials, we have designed a 4-point bending setup, which is now being fabricated in our mechanical workshop. So in principle, in this setup, the sample is clamped at the two extremities, and we apply a force in the 2 points near the center of the sample. So once we bend the sample, there is a finite strain induced both in the substrate and then transferred to the 2D material. So while this is still being fabricated, I have thought of some easy experiment. So trying to cover some micropillars, some polymer micropillars such as Ormocomb and PDMS covered with the WS2 single layer. So we can see on the top view, we can actually see that the material can be even suspended between various pillars. We can also see these little triangles, which corresponds to different number of layers. And if we see from a tilted perspective, we can actually see that the monolayers cover very nicely the pillars. So these are just some preliminary experiments just to see how can we measure strain in such curved WS2. And we do it mainly by means of the photoluminescence. So here's an optical image of one of these pillars. So you can see it's partially covered by the single layer WS2. The diameter of the pillar is about 5 micrometers. And in the PS, see, first of all, the neutral exciton A mode. And we also observe a little presence of the trion peak. Using the PL, we can make a map which corresponds to the wavelength, the change of the wavelength as a function of the position. And using some previously reported data, which relate the position of the peak to the percentage of strain, we can actually calculate the strain field of the 2D material on top of a micro pillar. So here we see the optical image and the corresponding strain field, where the white circle roughly corresponds to the pillar edges. So we see the majority of the strain is actually located in the center of the pillar, so that's about 0.28%. And that is where the 2D material is sort of like anchored on top of the pillar, and then the strain slowly relaxes as we move towards the edge of the pillars. So at least we can measure the photoluminescence as a function of a position with the resolution of approximately 500 nanometers. And we can use these maps to convert them into strain maps using some previously reported results. So of course, what would be interesting is to measure the PL wavelengths as a function of controlled strain on different substrates and for varying stacking. But for that, we have to wait for the 4-point bending holder. So for the conclusions, I would say we have successfully developed sulfur-based growth of the 2D materials and their transfer. We have measured the effects of substrate confinement and the effects on the exciton trion energy. And we obtained some first quantitative measurements on strain distribution in WS2 micropillars. So what is still missing is, of course, the dynamic strain studies, time-dependent measurements, and the measurements for heterostructures. And another interesting thing I think would be also development of the selenium-based TMDs, so molybdenum diselenide, tungsten diselenide, and their heterostructures. So with that, I would like to thank my colleagues from the photonic and phononic nanostructures group, Professor Clivia Sotomayor-Torres, Maria Jose Splandiu and class, and all the people that are collaborating with me on this project. And of course, I would like to thank the Severo Ochoa ICN2 Equal Opportunities for giving me this opportunity.
Thanks, Mariana, for the nice presentation. I don't know if there is any question in the room, in the seminar room. No? Okay. I think there is no questions in the chat. Oh, there is one question in the chat from Neus Domingo. She asked how you estimate the strain.
How do I estimate the strain? So in this really small experiment that I'm showing now, what I measure is the position of the PL emission, and then we use some results reported previously in order to convert the PL wavelength to the strain. But in principle, the idea also behind this project is to be able to do this calibration, so the wavelength versus strain for different materials and for different substrates.
Thank you. That's interesting. I think there was someone that took the micro in the room. There's a question there.
So can you hear me now? Okay, thank you, Mariana. Very nice talk. I had a question which is related to the title of your project, because you mentioned explicitly in the title electron-phonon coupling. So how are you going to address electron-phonon coupling with your experiments?
So the electron-phonon coupling is mainly related to the. Let me maybe go back to sharing my presentation. There are a bit more results that I did not manage to show because it's only 10 minutes. So in principle, the electron-phonon coupling is the coupling between the phonons and the excitons. So you can also see in this slide for the monolayers we do have a direct band gap. So we have the direct. So the exciton is corresponding to the direct band gap. But as we increase the number of layers, right? So for the bilayers and twisted bilayers, we will have more than one layer. That means we are already moving to the indirect band gap. So in order for the exciton to decay in the indirect band gap, we need the help of a phonon. So that's in principle how are we going to study these effects.
I have also a question that in the last part of your presentation you showed the 2D layer on top of this PDMS template. Mm-hmm. And how much of this strain is related to the number of layers that are stacked on top of each other?
Yeah, that's a very, very good question. So I only have like good results. So I'm still analyzing the data. In the meantime, I managed to take maps of various different pillars. What is really easy to understand is when it's a single layer, what I have shown. But yeah, in fact, for the bilayers, we really observe. Yeah, well, the change of the PL wavelength is not so straightforward to interpret. So I'm still working on that. It's not so it's not so well understood right now. So
Okay, good luck with that.
yeah.
Okay, so I think we can move on. Thanks for your presentation. And we will go to the second part of this program. That's the previously awarded thesis. And first we will welcome Seren Chamur, who was awarded in 2019. So Seren, the floor is yours.
Thank you very much for the invitation. It feels amazing to be here after 3 years, really. I'm very excited. So my thesis title is Advances on the Synthesis of MOS at Scale, and I defended it in 2019. Thank you. So I want to start with the MOS and describing them. So these materials are cage-like structures that are produced by the assembly of metal ions clusters and organic ligands. And thanks to this hollow construction of its nature, they have amazingly large internal surface area. So that makes them very attractive for many applications. And because we can select any metal from the periodic table, and we can design countless amount of ligands, up to now there are over 100,000 documented MOS in Cambridge Crossover database. Okay? So there are endless of structures and design possibilities. These materials are quite versatile. So I gathered some headings from the random articles. There are some articles from popular science journals to articulate how exciting these materials are. So the first one is to cure cancer in drug delivery. So the second one is for hydrogen storage for green transportation. Carbon capture for the mitigation of mitigation of the greenhouse effect, and as a green absorbent for air conditioning systems. So they called it limitless application. They called it magic powder, MOS. And at the end, it came to saving the world. This was 5 years ago. Yet, we don't see still in the market these materials, and there are reasons for that. Although it is performing very well in the lab scale, why we don't see still in the market these materials. So there are two reasons, basically. Lack of volume production and lack of effective shaping methodologies. In my thesis, I covered both of them, but today I will focus on only the large-scale synthesis methods because I have only 10 minutes. So my project was a European-funded project, which is called PRODIA. So it aimed basically the development of large volume and cost-effective production processes for MOS. And what we did basically was we created water-based synthesis methodologies, because commonly used solvent for these materials is DMF usually. It is very toxic and very expensive to scale up. And green chemicals, by chemicals I mean the precursors. So I needed to find some cost-effective and benign metal salts, which are not chlorides and nitrates, because usually chlorides or nitrates are the ones that is being used. and the effective shaping methodologies, of course. So I use spray drying and batch processes to realize this transaction to the upscale industrial level to produce high performance material with a competitive price. Okay? So spray drying is a very valid technique right now in industry. It is widely used in food and pharmaceutical industry, so the transition will be easier. How we applied this to MOF synthesis was basically we had a precursor solution and we injected to the nozzle with a certain feed rate, and nozzle sprayed it basically with the as a micro droplets into the hot chamber, and every droplet acts as a micro reactor. With the effect of heat of evaporation, we created these MOF superstructures. This method worked very well with the simple structures like HQ1, copper palluble structures. But if you look at, for example, this structure, which is a zirconium-based MOF, which is called UO66 family, this zirconium hydroxo cluster couldn't be managed with this. Sorry, could manage to be synthesized this simple process, because this process is very fast and this structure is very complex. It needed more time inside the reactor. So what we did to solve this problem, we insert basically the silicon bath here to make process a bit longer, so that we promote the synthesis of this unit, the metal cluster, in here, and then we sprayed everything to make the assembly for the 3D structure, and it worked quite well. The previous spray drying synthesis was using zirconium chloride and DMF. I changed it to zirconium oxychloride, which is a more benign metal complex than zirconium chloride, and I couldn't use DMF, obviously. So I used water and acetic acid in the process. So I had some parameters to optimize, of course, when I changed to solvent and the zirconium salt. First two parameters are the parameters from the precursors, concentration of the precursors, and acetic acid concentration in the precursor solution. And the last three are from the spray drying technique. So I could increase the yield and the surface area by using this green methodology. And also, after achieving this, I extend this process to another zirconium family, MOF, which is zirconium fumarate, and it worked quite well. So the second set of material was HX1, which is a simple structure. So the previous spray drying synthesis for this HX1 was using nitrate, salt of copper, and of course DMS-based process. And I changed this to copper hydroxide. And I picked copper hydroxide because the process was only producing as a waste water, so it made very easy to scale up in the industry. So parameters to be optimized was concentration of the precursors, concentration of ethanol in the precursor solution, and the feed rate. And after the optimizations, I could increase the yield quite effectively with keeping the performance of the material. So we work with the partners, 12 partners, which are from industry and also academia. One of them was Exelon. We worked effectively together with the chemical engineers to build this spray drying tower. It was 12 meters high. I will show you some pictures to visualize it. So it was a huge tower with a huge nozzle. It was the size of me, basically. And we could produce 50 kilograms per day, and with the estimated price of 150 euro per kilogram. So basically, my thesis proved that by using green synthesis and water-based protocols, we can apply everything to the pilot scale and scale up the process. So thank you very much. I want to thank first, of course, my supervisors to make it possible, and on the up because they were supporting me always, and they were amazing friends. Thank you very much.
I have, first of all, congratulate you for the thesis, because indeed what you did is something really interesting. So you did a great basic science, but also you work on the translation of this basic science to the scale up of the particle, which is very much 50 kilograms per day is quite a lot. So and congratulations. So the point is that did you have to follow some regulations to scale up the production of the particle?
It was not that straightforward. First, I scaled up somewhere. I went to the chemical engineers, and they tell me, no, you cannot use this, you cannot use that. And I'm like, okay. I go back and change the procedure a bit. I mean, it was back and forth all the time. It was teamwork, basically.
And it's very important to design the synthesis before starting the batch synthesis, because then you arrive to the scale up of the product, and then you realize that the reagents that you used before you cannot use because they are toxic, because they are not green, because they have side products that you cannot use. But it's very interesting, because you move from the basic science to the development of the scale up of the material. So congratulations, Chere. I do not know if there is some questions in the room. If not, Sonia from the chat.
Yeah. So there is no questions from the chat, but yeah, I would like to ask her one question. First, yeah, congratulations, because upscaling something that we do in the lab to the industry scale, it's something really challenging, and you did it. Yeah, that's very impressive. And I see that you tried to upscale 2 different MOFs, but did you find any common optimization parameters that can be applied to other MOF types?
We can, yes, extend the procedure, but if it is totally different metal, then it's a different story. The optimization should start from the beginning.
Okay, I see. So there's a lot of work left to do. So, well, thank you again for your presentation. I think we will move on to the next speaker. So the next awarded thesis, it's from Enelia Cristina Peláez from last year, well, from 2020. So welcome, Enelia.
Okay, good morning, everyone. I'm glad to see you again, and I'm going to present a summary of my PhD thesis called Nanoplasmonic Biosensors for Clinical Diagnosis, Drug Monitoring, and Therapeutic Follow-up. Our greatest motivation is to improve the quality of life of patients. There are current conventional methods, such as ELISA, PCR, that are very effective to obtain reliable results. However, there are several limitations to monitor self-control of a disease or to decentralize in laboratories. For this, we wanted to contribute with the development of point of cares to overcome those limitations. According to the World Health Organization, there are a set of criteria that called ASSURE to guide this development of these biosensors, these point-of-care devices. Biosensors is the main technology that can fulfill those criteria
Biosensors are three elements: the bioreceptor that interact with the specific analyte of the samples, a transducer that convert the chemical interaction in a electrical signal, and the signal processing system is for the easy visualization of the signals. Within the transducer, the optical, there is it has recognized like the main, the high sensitivity for those to become point of care biosensors, point of care devices. The optical biosensors are based on the evanescence wave. These evanescence waves are structures that confine electromagnetic waves into a guide mode or waveguide. When it's excited in the external medium, it generates an evanescence wave, and this can monitor with the light properties with many changes in the evanescence in field. Within this evanescence wave biosensor, the most representative is the SPR, the surface plasmon resonance. In my work, I have worked with the surface plasmon resonance and the localized surface plasmon resonance. In the first, we use the propagate surface plasmon with a thin layer metallic surfaces because there are many advantages such as versatility, robustness, and a high sensitivity. Then, to improve the miniaturization and we obtain a high surface sensitivity, we use metal nanoparticles with the localized surface plasmon to improve those conditions. Our group have designed an SPR platform. In this case, as you can see, it has a trapezoidal platform. Inside we can put the sensor chip, or the optical system, and the microfluidic system to inject samples. These signals pass to a spectrometer. We have worked with gold nanodisk structures and the thin layer of gold, and our group made a homemade readout software to see the signals in real time. We evaluate all the SPR sensorgrams in which we monitor the wavelengths, the minimum of the wavelengths, with the displacement to refractive index changes. So, for example, we can monitor the real-time tracking of signals when immobilized bioreceptors or to detect the analytes. An important process of this work was the optimum analysis. In this, we obtained the key factors to the surface biofunctionalization, such as the strategy for bioreceptor attachment, the density of bioreceptor, the accessibility of the target, to find the spacers, to avoid the steric hindrance, to obtain the functional group, to covalent binding the bioreceptor as a stable linkage, the orientation, and to maintain all the structure and functional property of the sample. Finally, we obtained the best strategy, the assay format, to detect these analytes. And finally, we found the antifouling properties because at the end we used biological samples, but it generated a lot of non-specific adsorptions on the surface, so we needed to find blocking agents to prevent those non-specific adsorptions. In my work, I have worked 4 types of biosensor methodologies, and in this case, I'm going to talk about, with a brief, only 3. Okay, for the first one is the anaplasmonic biosensor to monitor the acenocumarol. This is an oral anticoagulant that is needed to administer those needs to be adjusted for each individual. The goal is to self-control for the patients. So with this, we made an indirect competitive immunoassay. We obtained all the assay conditions, and we work with the effect of the plasma matrix using the strength sulfate as a blocking agent. And as you can see in this graph, we can eliminate non-specific absorptions of the plasma. So we obtain calibration curve with analytical parameters that we obtain a high sensitivity of the assay. In here, the working range is within the range report in real samples, and we obtain a study of accuracy with a 92 and 103%. The other project that I work is a plasmonic biosensor for the monitoring of gluten-free diet directly in human urine. In this case, the celiac disease is a chronic autoimmune disorder induced by the ingestion of gluten proteins. So there is no cure, there is no treatment available, but the only effective therapy is the gluten-free diet. On worldwide incidence, we can see that it's an underdiagnosed disease because 85% of cases are not still diagnosed. So the main problem is that the low adhesion to the diet along the life. So we try to develop a biosensor with the gliadin. The gliadin is a protein, the main protein of the gluten. When it's digested, it generates gluten immunogenic peptides. The main toxic peptide is 33-mergliadin. So we made an indirect competitive immunoassay to detect the 33-mer peptides in undiluted urine with 2 antibodies. We made all the conditions. We studied the undiluted urine matrix effect to eliminate all the non-specific adsorptions. We made all the calibration curve, and we obtained that the G12, the antibody G12, was the best sensitivity in this case. Okay, then we validate with patient samples. We use a commercial product that was a semi-quantitative. So we obtain excellent precision, and this is a monitor for 2 patients when we induce per day a little concentration of gluten, and we can obtain the concentrations of the GIPs in nanogram per milliliter. This is the first quantitative analytical tool for gluten intake control using undiluted urine samples for celiac patients. Finally, this project is for the detection of tuberculosis. Tuberculosis is an infection that caused by Mycobacterium tuberculosis. 1/3 of the population has this in a latent stage. So there are a lot of diagnostic biomarkers to detect in latent stage, but there are a lot of disadvantages because it's a variable sensitivity and low specificity. In our case, we obtained 2 different biomarkers, but I'm going to show only the heat-shock protein because it's a potential for early diagnosis. In this case, we only made a direct immunoassay. We obtained all the calibration curves. And finally, we studied with pretreated non-tuberculosis sputum samples to finally obtain the accuracy with the sputum in 97 and 103%. We made the validation with patients. With the samples has bacilloscopy positive or samples negative bacilloscopy, and we obtained that there is significant statistical differences between tuberculous and non-tuberculous samples. Finally, we made a raw curve with excellent precision. This, as a conclusion, this is the first report biosensor to quantify heat-shock protein in sputum. And this biomarker in sputum is also a first reported. And that's all. Thank you for your attention.
Thank you very much, Cristina, for the amazing work. So you work with a lot of markers, with a lot of diseases, and with a lot of matrix. So congratulations, and it's impressive, honestly. I don't know if there is any question in the room. If not, I have one question. So how is this for you? How is it was for you to translate the conditions optimized for one disease, for one marker to another marker? Because I assume that the matrix is different, the antibody or the marker that you use is completely different. So how did you make this translation straightforward in a thesis you managed to work with all of these diseases?
Well, yes, it's a little difficult, but for example, we have found different biomarkers, and we needed to try to interact with the specific antibody. So we need to play with the specific monoclonal, polyclonal, and the analyte can get a good detection, because, as you know, there are many samples that you need a label to improve the signal. So in this case, we could work exactly the matrix exactly. Yes, we have found different types of blocking agents. I have a list of blocking agents in my thesis. Yes, and the best it was that. Go on.
Okay. Congratulations.
Because you did an amazing work, and it is for sure in your PhD dissertation. Sonia, I do not know if there is any questions from you, from the chat.
I see there's no questions from the chat. Yeah, I also want to congratulate you for this amazing work you did. Very, a lot of development.
I do not know. If not, we should move to the next part of the talk, which will be by Kumara. No, Thank you very much. The previously awarded publications, and the first one is going to be by Kumara Cordero, who was awarded in 2019.
So thank you very much. Be here, and I will show you a briefly summary of the paper that it was awarded a few years back, where the objective of this work was to study the effects of flexoelectricity on the fracture properties of ferroelectric materials. So for those that are not familiarized with the concept of flexoelectricity, flexoelectricity is basically the coupling between polarization and strain gradient. And the more intuitive way to understand this is we consider a homogeneous deformation, centrosymmetric structure, and we apply a homogeneous deformation on it. We can clearly see from the sketch that the central symmetry remains, and therefore no polarization is induced. But when we apply a homogeneous deformation, we can clearly see that we break the impression symmetry, and therefore we induce some polarization to the lattice. Now, why this property is interesting, or one of the reasons that this property is interesting, is that if we can see from the equation, is that it's described by a four-rank tensor. That means that is allowed in all kinds of symmetries, so it is a universal property. Moreover, flexoelectricity is proportional to the permittivity of the material. So the effects of this property will be really big in ferroelectric materials that are well used for applications. And also because strain gradients scales inverse with the size at the nanoscales, this effect will have a big effect, and it had been shown that this effect can be comparable or even be higher than piezoelectricity. And because ferroelectric materials are also piezoelectric, it is really important to see how these two properties interact when they are present in ferroelectric materials. Now, an ideal source to study flexoelectric effects is or are cracks, because the front of a fracture
is the highest local strain gradient that we can have, the highest deformation. So we will have a lot of flexoelectricity there. It had been theoretically predicted that, for example, in barium tintanate, the cracks can induce a polarization that is 10 times bigger than the spontaneous polarization of the material. And because of this, or as a consequence of this induced polarization, the fracture toughness in this material has been shown to be asymmetric with respect to the design of the polarization. Now, the reason behind this asymmetry, or we can understand this asymmetry if we consider the energy cost of deforming a ferroelectric material, because it has two components. One related with the deformation itself, that is the elastic part, that is this here. And then the other is the electric one related with the induced polarization. Now, as I said before, if deformation is homogeneous, both will be symmetric. But as soon as we make an homogeneous deformation, what is gonna happen is that we have two sources for the induced polarization. One coming from the flexoelectricity, and the other coming from the piezoelectricity. And because the piezoelectricity depends on the signal or the sign of the ferroelectric polarization, these two can be acting in the same direction or in opposite directions. And therefore, the induced polarization that we will have will be different if the polarization is up or down, and therefore the energy cost will be different. Now, based on this, during my PhD, we demonstrate in uniaxial lithium niobate ferroelectrics that, in fact, plastic and elastic properties are asymmetric. And what we observed was that because the upward domains have a lower contact stiffness response, they are more flexible than the downwards. But at the same time, because they have a smaller plastic energy deformation, or they present a less plastic energy deformation, they are tougher than the downward domains. Now, motivated by this, and in order to experimentally prove that the fracture toughness is asymmetric, we perform some indentation, but in this case, Beaker indentations, and we use single crystal, periodic pole single crystal RKTP with the polarization in plane that is parallel to the surface, so that we can have cracks propagating along the polar axis. So we will have cracks propagating parallel and anti-parallel to the polarization. And also we will have, with this type of indentation, we will have cracks that are propagating perpendicular to the polarization. These ones we will be like a control way to see that our results are robust. So to extract the asymmetry on the crack length, we use this formula here. And the results show that in the case of the ones that are perpendicular to the polarization, we don't see any asymmetry trend, which was something that was to expect, because in that direction there is no competition or no interaction between flexoelectricity and piezoelectricity. But when we look at the cracks that were propagating along the crystal, the polarization, we observe a clearly trend where the cracks that were propagating parallel to the polarization was longer. Now, if we look also at the fracture toughness, that is the resistance of a material to propagate the cracks, we observe that when flexoelectricity and piezoelectricity are antiparallel, this fracture toughness is enhanced. So this is the explanation, or this confirm that, or this is the reason why we have that when we have different lengths for cracks when they are propagating in one direction or in the other. So in summary, what I have shown you is that the propagation of cracks depends on design of depolarization in ferroelectric materials, and that the toughness of ferroelectrics is like their polarization switchable. With this, I want to thank the co-authors of this paper, especially Professor Gustavo Catalan, and you for the attention.
Thank you very much, and congratulations for the work done.
Thank you very much.
I don't know if there is any question in the audience. I have one question. So what was the outcome of the paper? I mean, usually after publishing a paper, there is always a remaining amount of work that either you do not publish or you consider it strategic to publish later on. How was the evolution of this paper? So what's...
I didn't show other things, but first, this paper was published after I finished my PhD. In fact, I was already in Geneva when this became published. And then, for example, we estimate, because as I said, we can induce some amount of polarization, right? And what like after this, we compute the flexoelectric field that we can be inducing, and we observe that, for example, in the case of KTP, the amount of flexoelectric, the field was not enough, or it was too small to switch the polarization. But we, in fact, use another one, another material, and we saw some switching, local switching, because of the propagation of the. I mean, I have the slide. If they put it again, I can show it. So we demonstrate that that is not only that cracks propagate differently, but we can manually, we can locally switch the polarization of our single crystal, which is really difficult, by just crack propagation. So that was another output from the paper. But because of the 7 minutes, I didn't put it.
No, if it's Sonia, there is any question, or you have questions, or there is any question in the chat?
Congratulations for the nice talk. Yeah, there is no questions in the chat. I had a very general, sorry, I don't have it. Okay. I have a general question that maybe you explained at the beginning, but I don't remember. Which applications do these materials have? It's like energy related applications?
Also as memories. So they are used for memories and transducers, basically, and sensors, principle.
Okay, thank you. I see.
So thank you very much again, Kumara. And let's move to the last talk of the day, which will be by Maria Soler. We won the best publication in 2020. Thank you very much, Maria, to be here, and the floor is yours.
So hello everyone, and thank you for inviting me to this talk. I have to say that this was a review published in collaboration with Professor Andrea Armani in South California. We were talking about generic photonic solutions for COVID-19, but in this talk I will focus on our topic, in this sense, that is next generation healthcare. I guess I do not need to explain again why point-of-care biosensors are useful in diagnostics. We bet for them because they can improve, simplify, and among all, accelerate the diagnostics and the healthcare flow. And we use the biosensors. Already Cristina and Maricruz explained why we use these biosensors. So we use these photonic biosensors because they are label-free, they allow real-time, they allow to quantify, that it's something very useful in many aspects. We offer a very high sensitivity, and one of the main assets is the versatility. We can apply these biosensors to almost any type of biomarkers, like proteins, nucleic acids, cells, whatever we can think about. So in our group, we work with two types of biosensors, nanoplasmonics and silicon photonics. Christina was talking about nanoplasmonics ones. Cruz talked about the silicon photonics. But we don't do only this research line, we do the whole road towards the application and implementation. We do the device engineering, and we also do the developing of all the bioanalytical applications, including surface chemistry and assay validation. So as I said, we have two main devices in our lab. One is the SPR biosensors, or nanoplasmonic biosensors. Right now we have these SPR prototypes in our lab. We have up to 8 or 9 parallel devices working routinely in the lab. And in the case of the photonic biosensor, we have this type of devices. They are in a more laboratory scale right now, but we are also working on the integration of this technology in some kind of prototype and for subsequent industrialization and commercialization eventually. So we have worked, and as you can see, in a lot of applications, including proteins, nucleic acid cells, and pollutants for many diseases. But since the pandemics arrived, we got this project, and we focused on this coronavirus detection. So I will talk about mainly what we did in the COMBAT project. You may have heard about this. This is the project that we are coordinating from here in collaboration with the Marseille University and the INMI in Rome. And what we want to do here is to, I don't know, no, I cannot play the video, but what we are doing here is to develop this photonics nanotechnology for the detection of SARS-CoV-2. But we do not expect that to only be applied for human diagnostics, but also for monitoring of beta coronavirus or other type of coronavirus in reservoir animals in order to monitor their evolution and to prevent from other pandemics. So what we planned for this project was to develop our platforms, to apply our platforms to all what we can think about that it's possible. So we can detect the direct virus in a very similar way to the rapid antigen test that we all have used, but with the difference that we can quantify the viral load, and we expect a higher sensitivity to these lateral flow assays. We can also address the genomic analysis, but in a PCR-free way. So we do not need any laboratory conditions or any amplification that will lengthen the process, sorry. And we could use this as a very highly specific detection of SARS-CoV-2 viruses, but also we wanted to implement a multiplexing capability to be able to discriminate between COVID virus and flu and other common colds, for example, or for the monitoring of animals with this strategy. And eventually, and it was not planned in the project, but since we have the technology and we have the knowledge, we also went for the serological assay that it's about to detect antibodies in blood serum. And this is, we believe, is really important, not only as a retrospective diagnostics, but it could be useful for evaluation the patient immune response and the acquired immunity. So now we are all going for the booster dose or vaccines. So why not having a personalized test to know when our antibodies are diminishing and where and who and when is needing a booster of the vaccine. So this is what we propose with these serological assays. I will summarize the results we got so far. First, I will start with the later, that is actually the first one that we got out. For that, we use the SPR biosensor, and we mobilize on the sensor a combination of the viral antigens, the spike protein and the N protein that is inside the virus. And with that, we could improve a lot the sensitivity and the specificity of the assay by combining this multi-target approach. We directly went for real sample analysis These are positive and negative samples that we got from in collaboration with Weidebron and Clinic Hospitals. And seeing these good results that we could distinguish very well, we went for the full clinical validation of the technique. We measured more than 150 samples, and we obtained this sensitivity of 99% specificity of 100%, demonstrating that was much better than lateral flow assays, and even better than some ELISA and CLIA that they were using at the lab at this moment. In the case of the genomic analysis, what we are trying to do, as I said, is this multiplexing approach. We will immobilize the probes targeting different genes of the coronavirus in our sensor, and just by breaking the virus with some solution, we can directly flow the virus and detect these specific genes in our sensor. And the preferred application, let's say, is the direct detection of the virus. What we are trying to do here is the direct detection and capture of the whole viruses, not the proteins. So we are using some nanobodies that we obtained in collaboration with the CMB in Madrid. And this is what we are getting so far. We are detecting the viruses at the concentrations as low as 50 of 100 viruses per milliliter. So if you think of SARS-CoV-2 patients, the concentrations are around 10 to 3, 10 to 5. So we are well below this clinical cutoff, and we are trying to monitor. We are assessing the performance of these sensors with different variants of the virus. We have proven the good detection of the original one, the European, the Brazilian, the UK, and now we are also trying with Delta and Omicron in the lab. And we hope to finish this summer with some clinical patients that we will validate in Italy. As a bonus track, okay, just that we are also working on monitoring or evaluating some therapies, antiviral therapies for the COVID, and this we can do it as well with our sensors. And yeah, with these results and with all the past results that we have showed, this is what we are doing now. We are initiating the technology transfer, because there were some questions about the patenting and so on. Yes, we are trying to initiate this technology transfer and start a spin-off soon to commercialize all this. And thank you very much. And of course, thank you to the group that were a very female group.
I have a question. I don't know if there are any questions from the audience. If please, Pablo.
Yeah, thank you, Maria. So I have a question about the genomic analysis that you mentioned. So can you distinguish different variants of the virus with this genomic analysis by changing the anchoring group?
Actually, with the genomic analysis, what we are targeting are the more conservative regions of the virus, so do not have the problem of the variants that we have with the antibody one. So, but eventually, if we target the genomes or the genes that are supposed for the S or the RBD, yes, we could, but we are targeting the opposite. So to be able to detect all the SARS-CoV-2 independently of the variant.
I had another question, but very brief, Maria. How you work when there is no state of the art in the topic? I mean, we were always trying to write a review or write a paper, and then we have a lot of information on the topic. How you did it?
Well, it's the first time that we work with SARS-CoV-2, yes, but there is a lot of state of the art of biosensors applied for viruses. So for flu and influenza, there are hundreds of biosensors developed. For other coronaviruses, like the human coronaviruses, and we have also experience with bacteria, with proteins. So it was not that difficult to start. It was more difficult to get the reagents, for example, to get the yes. Well, actually, it was commercialized because when we started already in China, they had.
Okay.
But then we found a lot of problems with the reproducibility of those commercial antibodies and so on. So, yeah, that was the.
Okay, great, thank you very much. I don't know if Sonia in the chat, is there any question there?
No questions in the chat. And actually, the question I wanted to ask was similar to the previous question about how to distinguish the different COVID variants, but I think that was addressed. So yeah, there's no further questions from me. Just like congratulate again for the nice work. It's very impressive. And it's very exciting to see that you are starting a spin-off on this technology. Yeah, congratulations.
Much, Sonia, for your for to be chair of this session. Thank you very much, Maria, and all of the speakers. Now we just switch off for 2 minutes, just a pause of a break of 2 minutes, and we will come back with the round table and a new configuration of the room. Thank you.
Okay, welcome again to the Women Talent Program final event. Now we are in the round table. We have all the speakers and members of the commission here. Cruz, Maria, Nuria, Adriana, Seren, and Kumara. And we are going to debate a little bit on the topic of the effectiveness of the tools for the promotion of women in scientific careers, and in particular, what do you think of the ICN2 Women Talent Programme? And first of all, I want to ask you, Nuria, that you have been part of the commission for the last years, and you were at the very beginning when you decided to launch this program. What was the motivation of the program, and how you did it, you and all the members of the commission?
Okay, thank you to all to be here and for letting me. We have this couple of minutes to explain this. So the aim of this program was on the analysis of the results of the first Equal plan of the Institute, and there we realized that we had a not bad number of women starting their research career at the Institute, but that we were lacking women on the coming phases of their career. So we wanted to put in place a tool, an instrument that helped us to boost their careers, to maintain their profession on the research field. So this is why we thought that we needed to make their CVs like bright among the others by some tools that can make their curriculum more brilliant when they apply for other jobs or for other competitive calls or whatever. This is why we choose to boost PhDs and junior postdocs careers with these awards for their papers, their theses, and to fund some small projects to bring the opportunity, as Adriana said, to be PI for the first time in a research program. And really, I am very curious to know what you think about it, because now this is the moment for us to evaluate the program and to see what can we do forward.
Yeah, definitely. I thank you for this opportunity to share my thoughts about the program. And I think, it's been a very positive experience for me to being able to be a PI of a project. For us as postdocs, it's not easy to become PIs or to demonstrate these management, let's say, or these management skills, not skills, but this management opportunities to guide a project or to manage one. So I think this initiative of awarding these small projects, I mean, doesn't need to be a huge amount of money, but at least you have the opportunity to demonstrate that you're able to manage and to work as a leader of a project, of a small project, is very positive. I mean, not only to gain these such skills, but also to demonstrate that you're able to do so, and to improve your CV. And for me, for whenever I apply to positions outside ICN2, has been valued and acknowledged very positively. So I think it's great to count on this initiative here at ICN2, and I really appreciate it, and I really thank this opportunity. So I think it's something that should continue.
Adriana, you explain us all the benefits of the program, but I want to see from Cruz a little bit, because we won another project, and what can we do better? I mean, what can be improved? What would you need? What would you miss from this program?
Well, I totally agree with Adriana. Like, I mean, it's great for your CV, for your career development, and also for your motivation as a researcher. Maybe something that I missed, or maybe something that I would have liked, is maybe some follow-up, like not in 1 year, but maybe like every 2 months, or maybe to meet with the other participants in the program and have some networking events, something like we are doing today, but maybe like in a shorter period, just to keep the motivation going.
It is very interesting, because what you are telling us is something that we all need, which is the networking, no? To collaborate and to sit and to talk about our projects, our theses, our papers with a large community. So in this regard, the papers thesis later on, what do you think? So how do you think that was the problem? Did you realize it was intended for to recognize your work in the paper? What do you think? How it is?
So in my case, it was like a surprise. So I was not expecting it. I was not, since I finished, I didn't go away. But it was also really satisfactory to see that a paper in which you have work, it had been recognized, and also give you like a boost of motivation to continue doing it. Not because of surprise, but also because not because of the recognition or the prize, just because you can, that is telling, at least in my case, it was telling me that doing this fundamental research was appreciated, and it was important for the community. Apart from the community, was also important for women to see that we can have this kind of papers and also this recognition. So for me, it was like a boost to my motivation to continue working in academia. And I know, for example, that we make a joke with one of the girls from the group. I told, okay, I really win one, you should win one also, not like because they are friends. I think these kind of things motivate also the people that are here to work hard and try to get this kind of recognition.
Okay. And Maria, you are more senior. You won the senior award, so the postdoc award, you won the PhD award. What is the different impact in a postdoc career than in a PhD career?
Yeah, I will say that for me, winning a recognition like that for the CV is not very impactful. But I agree with my group that being here and having the chance to explain to other people in the institute what we are doing, what we are working with, and what we can leadership in these aspects is the the good feeling or the good response of this program. And I would like to say that I understand these recognitions of papers and theses as very important, but I think the major action of the program should be these projects, these PI projects that I think, because I did not receive one of those, but I received one of the Severo Ochoa Seed Funding Program, and I share this feeling that it really helps in the CV and in how they see you outside, that you already have been able to be a PI in a program, and this is your leadership capabilities are fulfilled with that. So, and this is very important for the career if you want to advance on it. So I think this should be the major action in the program.
Thank you. Seren, I don't know in your case of the PhD, so how was for you? What was the impact of anything?
It was a surprise, to be honest, because I was already in Cambridge and I was already working there as a postdoc. And it is always nice to receive an award and add to your CV. It was very nice feeling, and being back also quite nicely. I think these kind of things boost the self-esteem and motivation, as Mara said. It is very important.
I think that one of the it's that you say so. It's always nice to get a prize, no? And we all we all feel better with the prize. But beyond the let's say the contents of prize, do you think that this program or the awarded in this program, it's allowing to define role models of females doing a PhD, doing a postdoc, doing the leading projects? So what do you think? Do you think that you are creating, or did you represent some models for the?
Yes, of course. I think we are setting examples for the new generation, and it is very important what we do, our role here for the women in science, especially because it's hard to be a woman, as we were speaking before the event. But yeah,
I think it's a huge surprise, and it's a huge thing to celebrate that we are males and females here in the room, all celebrating the International Women's Day and the science that we are doing at ICN. And it's great to say and to consider that the science that you all, I include myself, so we are all scientists, that we all are doing is as interesting or even more interesting than the science that is doing by our male colleagues. But there is something that it came to pay my attention, is that we are coming from different backgrounds, you all are coming from different backgrounds, chemistry, physics, but I have the feeling that the science that you are presenting, some of you at least, it's more let's say it's more practical, so it's more towards the development of a product. And I wanted to share with you my impression. Maybe I'm wrong, maybe not. Do you think that the science that we are doing, it's more pragmatic, is more related towards? I'm thinking because in your case, in your particular case, you are the majority of the group is. What do you think about it?
Yeah, I think our research, in particular our group, is focused specifically in developing this product and this technology transfer. But I agree that the point where we are right now, with the sensors already up and running and everything, comes from a long research in basic research to discover these physical phenomena, to end up on how to fabricate them, how to characterize them, and so on. So yes, we maybe are at the end of the road, but we have to recognize that it comes from a long road in this sense.
Probably what you are telling us is that we are doing science in group, and the group it includes male and female, and the only thing that we are doing here today is celebrating that we are females doing science here. Nuri, I have a question for you. What was the what was the cons, the pros and the cons, but now the cons of the program? So what were the weak points of the program that maybe we will need to think about it, and we will need to try to improve a little bit in the future?
For sure. I think the biggest problem with this program was that the aim of the program was to enlighten your curriculum and to give you the opportunity to present the results that were awarded in international conferences. And as the very beginning of the moment as we started the program, the pandemic came, and we were not able to do so. So, but I am glad to hear that what you really wanted to do was being able to share your research with your peers, and this means that the objective, the main goal, was not so far from what would you expected to be doing, but for real, we need to think about doing an event like similar to this one we are doing today, but not every final program, but maybe every year, where we can share impressions and share your science for sure. And I really agree that we should put more effort on the projects that are really an improvement element for your curriculum, and for sure we need to improve our process. but this is yes, on our to-do list at the committee.
Okay. I want to open the debate and the round table to the audience. I don't know if you have any questions to the awardees. If not, in the meanwhile, I have another question. Is what's next? So we are talking about we have a Women Talent Programme final event, but what other measures we can implement to visualize, to make visible the science that the females at ICN2 we are doing? What would you think that it may be interesting to implement? I don't know if Nuria, you can
I mean, perhaps because it's a program that is inside ICN2, but perhaps it could be visualized, or like it could be like transferred, or like to shown outside ICN2, because I think it's very positive. I think it's a nice program that I don't know if it is implemented in many institutions. So I think it's nice to show how it is done here, and to I don't know, to show the results. I mean, to show that it's there are efforts here at ICN2 to promote the careers of women. So I think it would be a nice initiative to, I don't know, to do something, at least, for example, in BEAST. Start with BEAST and then to scale up to somewhere, because I think it's nice to show it outside as well.
Okay.
That would be my suggestion.
It's a very interesting initiative that you suggest. Amara, I don't know, Teren,
I think
what would you need? More money. We have to
I think what Adriana just pointed out is something that it is really important to do, because this way also, if we are more visible outside, probably more funding can become. That's a way to improve with funding, definitely. So doing that, what Adriana is saying, I think it's a good opportunity to bring more funding, and more funding means that we can, well, that the institute can hire more people, and more research can be done, and more females can be in the spotlight, and more people will be motivated and
I think it's very interesting what you point out. So well, first of all, I want to hear your opinion on it, and then, yes, please.
No, I mean, I agree with this. I was thinking not only to extend this to other centers, but maybe kind of trying collaborations between researchers from other centers, like these Ignite projects, for example, from this, but within women, I think that will allow us to share not only research, but also the mentoring, the feelings, or whatever. And we know that if we share this this motivation, we can help each other and advance in this sense. But we have to I think I'm being a pessimist here, but eventually, yes, this is helping our careers in the junior, postdoc, or senior, but then it's not all on us. And reaching the highest positions is not on us. We are helping, we are trying, and but this is something that we have to do. It has to be done with all the community and all the society, and I think just getting visible here and outside, helping students in masters, helping students in schools will also help to raise these female voices outside here.
I wanted to add something.
Yes.
One unexpected outcome of this program has to do with the evaluation of your research, of your papers, theses, and projects. And as you know, we recently signed the DORA Declaration. We are on our way to move forward to a more transparent evaluation processes and indicators and metrics, even if it's not the good thing to do. And when we started evaluating the papers and the theses, we realized that we needed to go beyond the traditional metrics for this kind of evaluation. And this is one path that we started with this program, but that will help us to evaluate the research outcomes in a different way, taking into account also this other difference, like also gender can be, because often female explains or create research outcomes in a different way than males. So we need to understand also these differences and being able to evaluate it as they are.
Mm-hmm. So I assume that then there are two phases, no? At least two phases. One is to analyze and to understand how the CVs or how the papers or how the thesis they should be evaluated.
Yeah.
And the second step is what you all, I think that you all put in place. It is so to make visible the science. And you talk also about networking. So this is the two different, the two things that you all consider as a common things that are important. In this regard, do you consider probably that the visibility is an issue when you try to develop a scientific career, or even more being female? Do you think that this is an issue? So we will need to work more on the visibility of the females that are working at ICN2.
I think this is a tricky question because ICN2 is visible because of the group leaders, and group leaders at ICN2 are mostly men. So I don't know how to respond to that in terms of ICN2, but in terms of scientific society, I think it's not about visibility, but there is a problem of self-confidence in this. For example, I have been collaborating with some programs with children, and one of the things that I see is that when you open the floor for questions, the boys are the first ones to raise your hands, and the girls are more shy, more timid. And if you give voice to the girl, then the others will come. And the same thing happens in conferences. When the floor is open, only male are the first ones raise their hand, and female are more shy, or I don't know why. Maybe I do it as well, but I think it's a problem of self-confidence, or how we are educated, how we are grown, and this is something that we have to change, not only from a scientific point of view, but in general.
It's empowerment, to empower ourselves to and I think that if I can add this, I think that I completely agree with you. So we need to think that we can do it. We can do it even better, I would say, but differently. And this is something that I guess that this is what you are saying. So if there is no more females than males, so you really is one. Please. Sí, perdona. I'm sorry.
I have many questions. Let me make just a couple of comments on the question. So one is about, so I understand that
what you like the most is the research projects, is the awards for the projects, right? So just a comment. So when we passed the midterm evaluation of the Severo Ochoa Award, we were criticized for that. They told us that we shouldn't be giving awards for projects just for females, that we should make it open, and that we should not make some positive discrimination. So I'm glad to see that the opinion of the majority of you is that the awards for projects is a positive thing. So that's reinforcing. Then the question is about something you said a couple of times. You mentioned the group leaders, and I think that's a key aspect of this of the success of this initiative. So I wanted to ask you, what was the let's say the reaction or the attitude of your group leaders towards your projects? So that goes for the awards for the projects, not for the theses and the paper. So what was the reaction of the group leader? Were they supportive? Were they say something like, that's a side thing for the group? Did you get full autonomy to work on it independently and by yourself? I guess that has many answers for each of you. But what was your general impression about the reaction of the group leaders?
Well, for me, it was positive. I mean, it's my group leader is me, so it was very positive and supportive to count on on service and class support and help. So they were very. I mean, still because as I mentioned, this is a work. I mean, a project that is still ongoing, and they have been very supportive, so I have no complaints about it. So, and also, it was very encouraging from them. I mean, from Sergio in particular, to me to apply for this initiative, to apply for this grant. So I think, in that sense, I really thank him and the group for their support. I have no complaints.
Yeah. So in my case, Laura was also very supportive and encouraged me to apply for it. I was also working in a project also related with the brain biomarkers. So it was nice also to find other applications that could be related also to the other project. I didn't have any problem with her. Yes, she was very supportive and gave me all the autonomy to work in this project.
Yeah, I guess my next question is whether this was a project that was considered as your project within the group.
Oh, yes.
If you got full autonomy to work on it independently, or do you have somebody watching over you?
No, well, it's as I said, it's a nice opportunity to control a project, or like to manage it, and then to be the head of a project. Let's say it was very nice. It was very positive.
Yeah, it was the same for me.
So I don't know if there is more questions in the audience. If not, I would suggest you to move the networking later on a little bit, because then we will run out of time. I want to thank, first of all the females here and the ones that they won the prize, but they couldn't be here for different reasons. Thank you very much for your time. Thank you very much for the great science that you are doing, and thank you very much to join us to celebrate this day, which I think it's very, very, very nice to spend some time, some morning doing science. And as I said, now as a chair, the only thing that I'm doing is taking over the work done by the whole commission. And as you may know, this commission was previously chaired by Neus Domingo, who now he wants to tell us a few words. And I think that there are...
So, hello everybody. So glad to be back at the Institute, even if just virtually, and to share this session with you. It was my pleasure to see all your faces here and to be able to follow up on the work that was awarded and that was founded by this Women Talent Project. But the most interesting part for me was this roundtable. So to confirm that some of the challenges and the aims on which we based and launched this program were kind of fulfilled. That's really rewarding. And I would like to emphasize a couple of things. You were commenting on how did we launch this project at the beginning. So certainly it was not very clear whether we had to or we didn't have to. It was one of the situations about, trying to decide what is more convenient, what should be worth, and how this could be controversial. And indeed it was, because in the first evaluation of the Severo Ochoa, there were some remarks against this program. But I think that part of the challenge was saying, why not? Let's try it. We do think there are some challenges we can really approach with this program. Let's try, and if not, we will correct it, right? So it was a matter of a little bit taking the risk, and I think that this risk was worth from all your opinions and from the outcome of this roundtable. It was really worth taking this risk, and I would like even to add some comments from the backstage. So it was, as you can imagine, just driving this project involves the participation of many people. It was also, an interesting learning process from the backstage on how to evaluate curriculums, on how could we, track which was the best paper, how to choose the best paper, right? So it was an internal discussion process that I think it was really enlightening for most of us about indexes, about how to recognize excellence, which is, it's a kind of debate that is always at the background, and that has a gender impact, has a remarkable gender impact. And we have to learn to play with some rules that maybe don't favor us, right? So part of this project was also, to learn on how to deal with that. From the backstage, I think that the best or the first outcome of this launching this program was something that I mentioned sometimes, and probably it will worth some deeper study. That it was not on the, I was feeling really sorry, kind of only being able to fund these really excellent projects with such a few amount of money, right? Which was part of the criticism as also that we received from the evaluators. But the main thing is that these projects, this Women Talent Program call, it happened to be launched kind of in parallel with the SEED project call, which some of you also applied. And there was one critical aspect that was really surprising us. And it is that there were as many projects presented for the SEED call as for the Women Talent Project, right? While in one call, we have 100% of the scientists of the institute that are able to apply, and for the other call, only women were able to apply. So the first question that was rising up is, since we have created two parallel calls, one is only for women, the only is for women and men, that's true. And probably that was the criticism also, where the criticism from the Severo Ochoa reviewers was arising. Since we have these two that are exactly the same, we are looking for exactly the same, the template was almost the same, and the evaluation process was, or the evaluation ticks were very, very similar, right? Why is there women that are not applying to the SEED call when it is obvious that they have the quality to apply for this Women Talent Project call? And there are different answers in this regard that need qualitative analysis in order to understand that. But after, one of these experimental fast checks among people who was applying, the main answer to this question was not, oh, the seed call is not for me. I'm just applying to the Women Talent Project call because this is meant for women. And so this is where I should apply. But indeed it was, oh no, I'm not good enough to apply for the seed project, right? So it's not like women were discouraged, the group leaders were directing women to apply for the Women Talent Project call instead of the seed call. It was a personal decision most of the times that were moving women to feel more confident on applying for a call in which, they feel more secure, they feel that they can compete at this level, and some kind of lack of self-esteem to apply for the seed project with exactly the same framework of project, right? And I think that this points to one of these questions that you have arised, that is the self-esteem, right? So I'm happy just to check that this Women Talent Project can be a playground where women can, develop skills by writing projects, by feeling that they can, or that showing that they can have leadership skills, but also showing the quality of the work that they are doing, and also playing with some of these rules that are very observed internationally, such as awards, right? We are afraid sometimes of awards. So, but internationally, the awards are something that are strongly recognized and relevant for CVs. So, of course, this will not be a award that will be internationally recognized, but it's a first step to train ourselves on the fact that these are important items to add in your CV, and also, to take into account, because now this is a small award, but there are other awards that come from international societies that, are relevant, that or that if you get some self-esteem on thinking that you can get them, the next step might be the relevant one that will have indeed the impact that is expected or desired in your CV, rather than this one. But still, that this is a first trial, right? This is a playground where to start developing these skills that then shall serve you for mid and long-term impact. So I just wanted to acknowledge and say thank you to all the people that has been able and has made it possible to continue this Women Talent Project. I know the effort that you have put, all the Equal Opportunities Committee, and I just hope, and this is a call for the management of the ICN2, I just hope that we can, consolidate. The beginnings are most of the times are the more critical part, but I really hope that after this Severo Ochoa program, Severo Ochoa 2, it can either consolidate on Severo Ochoa 3 or even be consolidated in the framework and as identity activity of the ICN2. And we should feel as that we have created this program. It's really interesting to think on having a networkship to develop this program also at the framework of BEAST, for example. But I encourage you to support this program for the future editions and also to adapt it and to make it evolve as necessary. But don't allow this program to get lost, because I think it's interesting, and the solution of continuity sometimes is what makes things big. So it is launched, we have created it, and now it's on your hands to bring it forward to consolidate it. And well, hello to everybody.
Thank you very much. Thank you very much, Ms. Severo. I guess it's quite early in the morning. So, and now that's done. Let's move to the last part of the event, which is the prizes, the awards for this 2021, which will be by Pablo, the fund director of ICN2.
We will also give some awards for the people that got the awards in the last years, the ones that have spoken today. So they will get a copy of this book of Laura Lechuga, a scientist, a women scientist jumping fences. So all of you will get one of these, and also the ones that got the award this year. So let me just go to the award ceremony. So first we have the winners of the best thesis award, which 2021. And actually we have two people that got the award ex aequo. So the first one is Tin Sang. So are you here? Yes. So Ting got her PhD with Jordi Arbiol and Juan Ramon Morante from IREC. The title of the thesis is Theme-Based Metal Organic Frameworks Derived Materials for High Efficiency Carbon Dioxide Electrochemical Reduction. So thank you very much. So congratulations. Yes. Oh, I gave you two. All right, so the second one is Maria Ruiz Garcia, who I believe is not here. I think we have the manager of the company that was involved in the thesis. No? Okay, Marta Ruiz. So, okay, so that's a thesis directed by Daniel Maspoc and Encarna Garcia Montoya from the company. And that's a design and characterization and optimization of novel liposomal-based food supplement against chronic fatigue syndrome. So that's Marta Ruiz. She's not here, so let's give her an applause anyway. All right, yes. Okay, so let's go to the best publication award 2021. That's for Noemi Contreras Pereda. Noemi, are you here? So the paper is entitled Synthesis of Two-Dimensional Porous Crystalline Materials in Simulated Microgravity, and it's on Advanced Materials of 2021. So, Noemi, here you are. Okay, thank you. Thanks a lot. Okay, and finally, the best publication for postdocs, that is Thais Grancha. I don't know if you're here. Not here? Okay. So the paper is entitled Synthesis of Polycarboxylate Rhodium-2 Metal Organic Polyhedra and their use as building blocks for highly connected metal organic frameworks. And that's, Thais is here. Okay, I can't see you, but. So congratulations, Thais. That's a paper on Angevante Chemie International edition into 2021. So here's the certificate for you.
While congratulations, Thais. While Alex is managing this, I just want to thank all the anonymous course referees that help us to develop and to evaluate all of these papers and theses. Thank you very much.
Thank you. So Alex is.
Can you hear me? Okay, because I cannot listen you. Thank you for this award. I'm very happy that this hard piece of work gets the recognition from you and that ICN supports the fundamental research. I also want to thank all the co-authors of this work because their contribution was key to develop it. And especially I'd like to thank my NanoApp colleagues. I miss you all a lot. And I'd like to extend the acknowledgement to ICN2 for creating this space and for joining these talented researchers. and give visibility to our works. So thank you so much, everyone. I miss you all, and I really would like to be there in person, but still I'm very happy to be here virtually. So thank you.
Thank you.
Bye-bye.
Okay, so let me just close the event. I just wanted to say a couple of things. One is that I think these kind of initiatives are really great to allow ICN2 to grow in equality and in the promotion of women in science. I think this is some key aspect of what we do. And certainly I think with the input that we got today, we will be able to improve the program in the next few years. So NEUS, don't worry, because this won't die here. It will grow in the next few years. And finally, I wanted to thank the people involved in the organization of all this, the Equal Opportunities Committee for starters, and NEUS for her really great work that she did during all these years. NEUS, not this one, but the one in the screen. We really missed you, and we wish you the best luck in the next few years in Oak Ridge. And also the other NEUS, which took the chance of taking over the work of NEUS Domingo, and she will keep working on this project for a few years, I hope. But also the other people in the committee. I see Josep Nogues here, but there were many other people in the committee working on this, and I think they did a great job. As Neus mentioned, the referees and all the people involved in the awarding of the prizes and so on. So thank you very, very much. And with this, I will finish and hope that.
Thank you, Pablo, to make it possible also.
That was
Thank you very much to stay here and have a
all right.
nice,
Yeah. So hope to see you next year with more people. I hope in the audience. All right. Thank you.